Hello everyone, welcome to Biotech Notebook. This is the platform where you can learn all biotechnology concepts. In this video, we are going to discuss how to calculate doubling time of microorganisms. So before going to solve the problem, first we should know the formulas to solve doubling time problems. Okay, so the generation time or doubling time G is equal to 0.693 divided by mu. So here mu is specific growth rate. Okay, so if the given data is specific growth rate, you can calculate generation time or doubling time by using this formula. Okay, and in the given data, if they have given the cell population or generation time, you can use this second formula, that is G is equal to T divided by N, where T is time interval and N is number of generations. Okay, by using this equation you can calculate N number of generation, that is B is equal to capital B into 2 power N. If you take logarithm of this equation, you will get the expression as N is equal to 3.3 log of small b divided by capital B. Okay, where capital B is number of cell populations before the time interval T. Okay, and small b is number of cell populations after the time interval T. So if you know these three formulas, you can easily solve the doubling time problem by substituting the given data in these formula. Then you can find out the required data. Okay, so let's see the problems. So the first problem is, in a bacterial growth experiment, the concentration of cells increased from 10,000 cells per ml to 30,000 cells per ml in 3 hours during the exponential growth phase. Okay, so here the given data is 3 hours, that is the time interval T is 3 hours, and at the beginning of your time interval your cell population is 10,000 cells per ml, that is your capital B. Okay, then after 3 hours your cell population is 30,000 cells per ml, that is small b. Okay, then they are asking the doubling time of the bacteria. Okay, so the doubling time of the bacteria is, so the options are A 2.5, B 3.0, C 1.0 and D 1.9. Okay, and this is a DBT 2017 question. Okay, in many life science entrance exams like DBT, JRF exam, ICMR exam, GATE exam and CSIR NET exams, repeatedly they are asking this type of doubling time problems. Okay, so the formula to calculate doubling time is G is equal to T divided by N. So here T is nothing but time interval in hours or minutes and N is number of generations time. Okay, so it defined as number of times the cell population doubles during the time interval. Okay, so here in the given data they have given time capital B and small b. Okay, they have not given the number of generations directly, so you have to calculate the number of generations first. So formula for calculating number of generation is N is equal to 3.3 into log of small b divided by capital B, where capital B is number of bacteria at the beginning of a time interval and small b is number of bacteria at the end of the time interval. Okay, so if you substitute this N in this equation, you will get the doubling time. So that is doubling time G is equal to T divided by N, that is 3.3 log of small b divided by capital B. So given data T is 3 hours, capital B is 10,000 cells per ml and small b is 30,000 cells per ml. So just substitute the given data in the formula. So you will get capital G is equal to T, that is 3 hours divided by 3.3 into log of small b. So small b is 30,000 divided by capital B is, that is 10,000. Okay, so G is equal to 3 divided by 3.3 into log of, so 30,000 divided by 10,000 is equal to 3, so log 3. Okay, so 3 divided by 3.3 into log 3 is 0.9053 hour. Then problem 2 is, what is the doubling time of a bacterial population that increases from 10,000 cells per ml to 50,000 cells per ml in 5 hours of growth. Okay, so this is for your practice, and just solve this problem and give the answer in the comment section. Second model, here we are going to calculate doubling time from specific growth rate. Okay, in the previous problem we are calculated doubling time from the generation time. Okay, so the problem is if the specific growth rate of the microorganism is 0.27 half power minus 1, find out its doubling time. Okay, so in the problem if they given the specific growth rate, then the formula is G is equal to 0.693 divided by mu, where mu is specific growth rate. So mu here they have given that 0.27 half power minus 1 and they're asking doubling time, that is G. So it's a very simple problem. So G is equal to 0.693 divided by 0.27. Okay, so G is equal to, that is 2.5666 hour. Next problem, the specific growth rate of a mold during exponential phase of its growth in a batch cultivation is 0.15 Harper minus 1. That is, your specific growth rate mu is 0.15 Harper minus 1. Okay, if the cell concentration at 30 hour is 33 gram per liter and the cell concentration in gram per liter at 24 hour is. So they are asking the cell concentration at 24 hour and they have given the cell concentration at 30 hour. So from this given data you can calculate time interval. So the time interval is T is equal to 30 hour minus 24 hour, that is 6 hour. Okay, so 30 minus 24 is 6 hour. Then here small b is the cell concentration after the time interval. So at 30 hour is 33 gram per liter, that is after 6 hour. Okay, 33 gram per liter, and capital B is the cell concentration before the time interval. So before the time interval is 24 hour. So that only they are asking. So from the specific growth rate mu you can calculate generation time. So the formula is G is equal to 0.693 divided by mu. So generation time G is equal to 0.693 divided by mu 0.15. So G is equal to 4.62 hour. Then you can calculate number of generation time from this formula, that is G is equal to T divided by N. So here T is time interval 6 hours and G is 4.62 hour. So N is equal to 6 divided by 4.62, that is equal to 1.3. Then you can calculate the capital B. Capital B by using this formula, that is small b is equal to capital B into 2 power n. So here B is 33 gram per liter, it is a given data. In capital B you have to calculate and n is 1.3. 2 power 1.3 is 2.5. Okay, so capital B is equal to 33 divided by 2.5, that is equal to 13.2 gram per liter. Problem number 5, E. coli was grown aerobically in a batch fermentation. The initial concentration of cell was 1 into 10 power 3 per ml and it reached 1 into 10 power 6 per ml in 10 hours. Calculate specific growth rate. Okay, so here the given data are, so time interval T is 10 hour and before the time interval the cell population was 1 into 10 power 3 per ml, that is capital B. And after 10 hours your concentration reached 1 into 10 power 6 per ml. So small b is 1 into 10 power 6 per ml. Okay, they are asking to calculate specific growth rate mu. Okay, so from this small b and capital B we can calculate n, number of generation. Okay, so n is equal to 3.3 log of small b divided by capital B. So n is equal to 3.3 log of b, small b is 1 into 10 power 6 divided by small capital B 1 into 10 power 3. Will cancel it out like 1 into 10 power 3. So you will get n is equal to 3.3 into log of 10 power 3. So 3.3 log of 10 power 3 is 3. So 3.3 into log 3 is 9.9. Okay. So from n you can calculate generation time. So G is equal to T divided by n. So T is time interval 10 hours and n is equal to 9.9. Okay. So G is equal to 10 divided by 9.9, that is 1.0101 hour. Using this formula you can calculate the specific growth rate, that is generation time G is equal to 0.693 divided by mu. So specific growth rate mu is equal to 0.693 divided by G. G is 1.0101. So mu is equal to 0.693 divided by 1.0101 is equal to 0.69 Harper minus 1. Okay, next problem for a microbial culture. If the doubling time is 0.231 hour, the specific growth rate will be. Okay, so the options are 1, 2.5, 0.3 and 3.0. Okay, so it is a direct problem. You can easily calculate the specific growth rate. So the formula is G is equal to 0.693 divided by mu. So mu is equal to 0.693 divided by the doubling time G, that is 0.231. So mu is equal to 0.693 divided by 0.231, that is equal to 3 half power minus 1. Okay, problem number 7. What will be the generation time of a culture with a specific growth rate constant of 0.01 minute power minus 1? So this problem is for your practice. So you just solve this problem and give your answer in the comment section. Okay.